Good morning, everyone. So we'll keep things moving today just to make sure we're keeping things on track. So next up we have Wang from the University of Hong Kong, who's going to be talking about the prevalence and transmission of antibiotic resistance in wastewater treatment systems.
Good morning, everyone. I'm Wang. I'm currently a postdoc in Professor Zhang's group at the University of Hong Kong. Before my presentation, I would like to extend my heartfelt thanks to the EDAR7 committee for providing us the chance to get together and share our research. Today, I will introduce our work, which explores the prevalence and transmission of antibiotic resistance in wastewater treatment systems by using metagenomics and high-C sequencing. Antibiotic resistance is one of the top global public health and development threats, as indicated in the report published by the Lancet. About 5 million deaths associated with bacterial AMR in 2019. It's important to note that antibiotic resistance can spread between people, animals, and the environment. So it's important to control the antibiotic resistance from any source. Antibiotic resistance hotspots were found not only in the medical settings, but also in some environment compartments, such as the municipal wastewater systems, which receive wastewater from different sources, including hospital, household, industry, and sometimes agriculture or farm. So wastewater treatment system is a double-edged sword that can act as a pathway for AMR spread or as a barrier to reduce the environmental release of AMR. So sensing the potential impacts of AMR on the subsequent environment is of great importance. Current research about antibiotic resistance mainly focuses on different aspects, including the identification and quantification of ARGs, and the transferability, evaluation, and host identification of ARGs. To identify the transferability and the host of ARGs is important for understanding their spread among bacterial populations and informing the strategies to control their dissemination. Currently, the popular method used for ARG host identification include culture-based techniques and culture-independent method. But we know the majority of the organisms found in the environmental systems cannot be cultured under the standard laboratory conditions. So the culture-independent method have shown their great potential in host identification. Here we compared the advantages and the limitations of some popular culture-independent methods, including the correlation analysis, metagenomics, cell sorting, single-cell sequencing, and high-C sequencing. Among these methods, high-C sequencing has shown its great potential in host identification. This figure, compared with the other method, high-C sequencing is non-targeted and high throughput. And the most important part is high-C sequencing is capable of linking the mobile genetic elements to their host. This figure shows the brief overview of the high-C sequencing workflow. The first and the most important step is cross-linking the DNA within the cell. In addition to the chromosome, other mobile genetic elements, including plasmid or fixed in close proximity, can also be linked. The cross-linked DNA is then cut with restriction enzyme, and then resulting DNA fragments are repaired and biotin labeled. Ligase is added to join the fragments from the DNA, and then which are then shared, which are shared and enriched and prepared for the high-C sequencing. Then the generated high-C reads can be used to determine the relationships between the genomes and other mobile genetic elements. So in this study, we aim to use metagenomic sequencing and high-C sequencing to detect the relationships between the ARGs and the hosts in wastewater treatment systems. In this study, we collected 4 different sample types, including influent, activity sludge, effluent, and anaerobic digestion sludge from 6 wastewater treatment plants. Subsequently, we would like to investigate the ARG profiles, the host ARG host identification, and the removal of ARG and ARB, the shareness of ARGs, and the potential transfer of ARG in the wastewater treatment plants. Okay, now I will introduce the preliminary result of this work. Firstly, we identified the ARGs by annotating the assembled contigs and detecting over 900 unique ARG subtypes from all samples. Then we explored the location of the ARGs by annotating the ARG carry contigs, revealing about 70 to 18% of the ARGs were carried by plasmid. So this result highlights the importance of linking the ARG carry plasmid to their hosts. By linking the ARG carry contigs to their respective host, we identified 416 ARG host associations in our study, which achieved 50% increase in host identification by using high-C sequencing. And we found hosts affiliated with Pseudomonas and Bacteroidota are the most prevalent. We also calculated the absolute cells of ARB in the samples per given volume. When comparing the influent and effluent, the lower ARB cells were found in effluent, achieving over 95% decrease in the effluent samples. For the AS and ADS samples, due to the sludge concentrated operation, more ARB were found in these samples. This observation implies the subsequent need for treatment for the excess sludge to remove ARG and ARB. By assessing the ARG, using the ARG host pairs, we found that more ARGs were associated some specific classes, including the alpha proteobacteria, gamma proteobacteria. And we also found multiple links of ARGs were prevalent in influent and ADS. We also found that many of the genomes linked to multiple ARG belong to the emoji pathogens. We also calculated the ARG copies based on the coverage of ARG host pairs, and we found that most of the ARG copy numbers were below 5. We further explored the shareness of ARGs and the potential transfer of ARG within wastewater treatment plants. We find the influent samples have more unique ARG, whereas activity sludge, effluent, and anaerobic digestion sludge, their ARGs were shared. We compared the hosts of the shared ARGs, and we found that many of their hosts were affiliated to different genera or species, suggesting the potential of horizontal gene transfer within the wastewater treatment systems. It's important to note that this result need more investigation in the future. We know in this study, the high-C sequencing has shown their great potential in ARG host identification. But we know high-C method relies on the short-read sequencing, so some important information may be lost during the assembling or the subsequent host identification due to the limit read length. So we hypothesize the POC method, which is based on the nanopore long-read sequencing, may be more powerful in host identification. So here to test the hypothesis, we first to evaluate the feasibility of the POC using a pure mock culture. And our findings revealed that the POC sequencing can help to assemble plasmid into the max of isolates within the mixture. And we further evaluate the feasibility and search for the relationships using the real environmental samples. And the POC sequencing generated over 8 gigabytes of data, and we found a rich set of links in the real samples. Currently, the part of this result is still being analyzed. Okay, in summary, high-C sequencing and POC sequencing have shown their great potential in host identification in environmental samples. Okay, that's all my sharing about this project. Finally, I would like to thank for the guidance from of Professor Tong Zhang and the help from my lab mates. That's all my sharing. Thank you.
Thank you very much for the very nice talk. I'm Ryo Honda from Kanaris University, Japan. So I'm also working on the antimicrobial resistome in the wastewater treatment system. And in the wastewater treatment system, there are two types of bacteria, the microbes from the wastewater origin and aerobic microbes, which are maintained in mostly in the activated sludge. And my question is, are there any? Did you find any difference in the ARG carried by the microbes of wastewater origin, which is the anaerobic gut microbes, and from the aerobic species, which is probably maintained in the active sludge?
Okay, thanks for your question. Actually, we found the influent samples have with the highest diversity of the ARG. Yes, for the you mean for the activities large samples?
Yeah. For example, the ARG of which drug classes is any difference between the ARG carried or drug class of the ARG carried by the anaerobic microbes from the aerobic microbes?
Okay, thank you. Yeah, we found the ARB was different from these different samples. For the AS, maybe actually the bacteria compared with the influent, less bacteria carried ARG. Yes.
Yeah, thank you.
One short question. How about the sequencing depth for your high-C sequencing combined with metagenomics sequencing? Because I found that there are more than 6,000 link pairs between the MGEs and the cells, right? So what's the sequencing depth for this technology?
Thanks for your question. For the metagenomic, the sequence depth is about 60 gigabits of data, 60 gigabits.
For each sample, right?
Yes.
Okay.
For the high-C sequencing, the sequence depth is about 30 gigabits.
Okay, thanks very much. Excellent work.
Thank you.
My name is Orestis Kanaris, and today I will talk about some key results from my PhD project, on the metabolic niches and the persistence of antibiotic-resistant bacteria in the environment. I think this audience does not need a big introduction on the problem of antimicrobial resistance and its importance, so I will skip the first slide. So people before me already started thinking in the direction of how metabolic niches can determine the coexistence or the exclusion of antimicrobial resistant bacteria in different environments. So I will show a short theoretical example from the literature and this way of thinking is also important for my project as well. So now imagine an environment where we have a sensitive strain and a background community and two substrates. So the arrows show which substrate can be used from which strain, and how thick each arrow is shows the competitive ability, for example, how fast these strains can grow on each substrate. And so now imagine two scenarios. So the sensitive strain becomes resistant, and it loses some competitive ability for both substrates, but it can still coexist with the background community because it can still use substrate 1. And in the second example, the sensitive strain becomes resistant and it loses the ability to use substrate 1. So now it has to compete with the background community for substrate 2, and it gets excluded from the system. The aim of my project is to better understand how the environmental conditions, and more specifically the available nutrients, can influence the selection of resistant bacteria. And we also want to see if we can select against the resistant bacteria by adding something to a specific system. So how we want to do this, first we wanted to isolate some E. coli strains from the environment. Then we wanted to characterize them for their metabolism and for their resistance, and then do some population dynamics models to simulate the competition of different strains under different conditions, and then perform competition experiments to see if our predictions are correct. When I started my project, I was lucky that in the lab we already had 62 E. coli strains isolated from wastewater treatment plants in Norway. And this was done by the working group of Dr. Nachiket Marathe, and they were provided to us. There is already a publication about how these strains were isolated and on their characterization for antimicrobial antibiotic resistance. And the two important things that you should keep in mind about these strains are that they were isolated on agar plates containing the antibiotic cefotaxime and also on non-selective plates. And they were already characterized for antibiotic resistance, and they were also whole genome sequenced. And this work was done by the working group in Norway. So here you can see the resistance of the 62 isolates to 14 different antibiotics. So here is shown the MIC relative to the clinical breakpoint of each antibiotic. And red colors show resistance, blue colors show sensitivity. And the main message from this plot is that most of the isolates are resistant to multiple antibiotics at the same time. And since I had the whole genome sequencing data available to me for these isolates, I decided to perform genome scale metabolic models to characterize the isolates for their metabolism. And this is what I did. And with the models, I simulated their growth in almost 300 different carbon sources in minimal medium, one carbon source at a time. And here you can see the result. So orange color shows growth and black shows no growth. And I was able to identify 40 carbon sources which could be utilized by only some of the isolates. And this is what I was looking for. But in the lab, I only work with three carbon sources, and these are this here. So firstly, dimalate, which keep in mind is not the same as L-malate. Dimalate does not occur naturally, but it is used in the food industry. And the second is sucrose, which is one of the most abundant carbon sources in the environment. And glucose was my control because all of my isolates are able to use glucose. And here I tried to combine the resistance data with the carbon utilization data. So I divided the isolates in 4 sections. So on the far left, that isolate cannot grow on either dimalate or sucrose. Then the isolates in the next section can grow on dimalate but not on sucrose. Then in the next, they can grow on sucrose but not dimalate. And in the last, they can grow on both carbon sources. And I noticed that in the third section, I had 5 isolates that were resistant to these 3 antibiotics and could grow on sucrose but not dimalate. And on the second section, I had 5 isolates that were susceptible to the same 3 antibiotics, but could grow on dimalate but not on sucrose. So in theory, if I would let these 10 strains grow together in a culture, if I would use sucrose as a carbon source, I would expect that I would select for the resistant strains, and if I would use dimalate, I would expect to select for the susceptible strains. So this is what I did. So as controls, I've let the susceptible strains grow on their own, and then the resistant ones as well on their own, and then all of the 10 strains together in minimal medium with glucose, dimalate, or sucrose as carbon source without any antibiotics. I've let them grow for 24 hours. And at the beginning, at the end of the experiment, I've spotted all the cultures on plates without any antibiotics and on plates with one of these 3 antibiotics to see the number of the resistant cells. And here is what I saw. So the susceptible cells growing on their own could grow on glucose and on dimalate and not as good in sucrose. The resistant strains could grow on glucose and on sucrose, but not really on dimalate. And then in the mixed culture, the resistant strains could still grow on glucose and on sucrose, but with dimalate as a carbon source, only the susceptible cells could grow. And with those data, I was able to calculate some selection coefficients, which basically shows that with dimalate, we were able to basically select against the resistant bacteria. But of course, in many environments, it is hard to remove a carbon source and to substitute it with another one. So we had this idea. So now imagine a setting where we have a resistant strain and a sensitive strain growing on one substrate, glucose, and they grow equally well, either because of compensatory mutations in the resistant strain or because of low levels of antibiotic compensating for any costs for resistance. So after some growth cycles, we would expect that we would have the same number of resistant and sensitive cells. But now we know that, at least in our selection of strains, the susceptible strains can utilize dimalate as carbon source, but the resistant ones cannot. So if we would add dimalate to this system, we would expect to have more and more sensitive cells, which would use more and more glucose. And after many growth cycles, we would expect that the resistant strains would the number of resistant cells would be less and less. So I tried to simulate this. So here is what I did. So I assumed deoxygrowth. So first the glucose is used and then dimalate. In the first growth cycle, I only had glucose in the system and then glucose and dimalate. And you can see that after each growth cycle, we have less and less resistant cells. But I did not do this experiment yet, but I will do it in the near future. So we But of course, we understand that what we do is just a proof of concept and that the conditions are much more simple than any environment, and that we only work with a selection of strains. And of course, if this would be applied, you would have to be sure what organisms are being boosted by the carbon source. But yeah, in the near future, we want to look for some more general patterns of carbon source utilization in resistant strains and work with more complex communities. Thank you, first of all, to my supervisors, Frank Schreiber and Jan Kreft, and to my colleagues at BAM. Thank you.
I'm Concepción Sánchez Cid. I'm in the University of Lyon. I'm a postdoc. And so I was thinking, in an environmental setting, for example, so you use E. coli strains, right? But let's say you have your environmental bacteria that are slow growers. So I'm wondering if you have an E. coli there and it's competing for the same substrate, would the exclusion be that much of a problem there? Because then it could use, E. coli could eat the substrate faster, right? So then maybe that wouldn't apply that much. Any thoughts on that?
So, of course, in real environments, it can be very different, but this is just a concept, and I mean, if this concept works, you could apply it for different organisms as well, so depending on your environment and the conditions that you are interested in, right?
Our next short talk will be given by Marius Surleac, and he comes from Bucharest, Romania, and will talk about clustering in the context, antimicrobial resistance and MGE dynamics in ESKAPE pathogens.
Thank you very much for the introduction. Yeah. My name is Marius Surleac. I'm a bioinformatician with a background in physics. And I work at the National Institute for Infectious Diseases in Bucharest, but also at the University of Bucharest. And I will share with you some of the findings that we have on ESKAPE pathogens from wastewater treatment plants and surface waters from Romania. But first, Romania is among the European countries with the highest incidence of antimicrobial resistance. And we have some, at least two serious problems with that. One of them is that the estimated population coverage is the lowest in the European Union, and usually, at least I'm not sure if there are too many surveillance projects in Romania. And another one is the high use of antibiotics for the population. Recently, in the last year, some governmental regulations have been made to force the use, against the use of antibiotics. And as you can see here, according to the last ECDC report on surveillance, between 2018 and 2022, there has been a 2- to 4-fold increase in species like E. coli and Klebsiella pneumoniae in Romania. For instance, there is a almost 4-fold increase for Klebsiella. We have sampled the isolates from wastewater treatment plants, the influent and the effluent from the treatment plants from hospital and also domestic and industry across Romania, and also the surface waters, the upstream and downstream rivers nearby the wastewater treatment plants. And for that, the isolates have been selected from 7 cities across Romania. These are industrialized cities and highly polluted. And we have whole genome sequenced 500 isolates from upstream, downstream waters and influent, effluent isolates from the wastewater treatment plants. And this is the distribution between species, the ESKAPE species of these isolates. These whole genome sequences have been used as has been used to predict antibiotic resistance genes, biocide resistance genes, virulence factors, and different mobile genetic elements. I use Diamond for that, and various databases like CARDRES Finder and CG databases and so on. And I will share with you some of the findings that we have on this data. First, I looked at the MLSE distribution between the most abundant ones. I will show only the most abundant ones that we have, Acinetobacter, E. coli, and Klebsiella pneumoniae. And here we can see that the subtypes that are worldwide distributed are also found in our samples, like subtype 1 and 2 in Acinetobacter, and also 101 and 258 in Klebsiella. In E. coli, the most abundant one is 167 in the Romanian strains. And if we look at the co-occurrence networks, we can see that some of the subtypes are shared between different types of isolates. For instance, in Acinetobacter baumannii, we have, for instance, subtype 1 and 646 that are present between influent, effluent, and downstream isolates, but it's not found in the upstream isolates. And similarly, in Klebsiella, subtype 101 is not found in the upstream isolates. Then I looked at the resistance genes that are shared or that are unique between isolates in this study. And the resistance genes are homogeneously distributed among species, at least it looks like that. In this co-occurrence graphs, you can see in the middle with purple the ones that are shared between all types of isolates, but there are also unique genes to various isolates. For instance, here are the unique downstream, the unique genes that were found in the downstream isolates for Klebsiella pneumoniae and so on. But we also can see that there are some other genes that are shared between, for instance, here between influent and effluent, or between effluent and downstream, and so on. Most of the genes here are covering most of the beta-lactam resistance classes of genes. And also we have aminoglycoside resistance genes among them. Also you can find CTXM 15 and 1 and 14, but also NDM1 and NDM5 among all the isolates from all the samples. Then if you look at the biocide resistance genes, most of the genes here are mostly efflux systems and heavy metal resistant genes, like they provide resistance to mercury, silver, and arsenic. But the co-occurrence networks look a bit different, at least for Acinetobacter baumannii. The number of genes that are shared between all of them is really low, and there are no unique genes that are found in upstream, and also in downstream we found only QSE. But for E. coli, there are plenty of resistant genes that are shared between all of them. And in Klebsiella, we can see a difference. There are plenty of genes that are shared to all of them, but a lot of them are shared between downstream and influent. If we go further, based on these predictions that I had, I wanted to see how these genes are related to one another, and for that, together with my student, we wrote a program in Python to look if these genes cluster together or not. And the clusters that we named are considered to be formed by at least 2 elements. And as you can see in this image, most of the genes are found solely in the genome, spread throughout the genome. But we also have clusters of at least 2, and then move further to get clusters of up to 50 elements. And elements here are considered antibiotic resistance genes, virulence factors, biocide resistance genes, mobile genetic elements, and even phages. And based on this data, I will share some of the clusters. So I split the clusters in a few groups. One group is for clusters that have less than 10,000 base pairs. The condition for a cluster to be formed is to have at least a maximum of 1,000 base pairs between each element. And we could see clusters of 2 elements. These are the clusters for antibiotic resistance only and mobile genetic elements. And we see different combinations of such elements from 2 to up to 15 elements clusters for this particular set. We can see here that we have various mobile genetic elements. We have ICE, we have transposons, we have insertion sequences, we have genes near the replicants from the plasmids. And where we have mobile genetic MGEs, this means that there are multiple predictions for that particular place in the sequence. That's why we have MGE and also IS and so on. And we also looked for antitoxin and toxin systems. But if we consider the relationship between biocide and antibiotic resistance, then we could see some combinations of those 2. And these are the antibiotic resistance genes that we found in such combinations. And these are the biocide resistance genes together with the mobile genetic elements between them. And if we go further, the bigger clusters tend to be formed by virulence factors and pathogenic islands and phages, and so on, for clusters that have more than 40 K base pairs that go up to 44 elements. And in conclusion, the MR dynamics in the environment is a major One Health concern, and we intend in the near future to sequence isolates from animals too. The transmission in the environment is not really known, and we think that the treatment in the wastewater treatment plants is not very efficient. That's why a lot of isolates are shared between influent, effluent, and downstream waters. And the context analysis shows a very diverse repertoire of elements. I want to thank my colleagues from the National Institute of Infectious Diseases and from the University of Bucharest and the funding, and thank you for listening. And
thanks a lot. And are there questions? Maybe I can start with a short question. You have mentioned plasmids a couple of times, but I haven't seen any plasmid and the name of it.
Yeah, I have one slide added here with the plasmids that we found. These are some network graphs between the plasmids that are shared between the types and in various species. And also, these are the genes that are found in the same isolate or in the same contig with the plasmid replicons. And most of them are giving resistance also to antibiotics. Vancomycin is the most abundant one, and also for heavy metals, mercury and silver.
Mm-hmm. So was this done with long-range sequencing or short reads?
Short reads.
Okay.
Illumina.
Okay. So.
Jan Siemens, soil scientist from Gießen. In your patterns of ARGs, I recognized that often the orange dot for downstream was closer to inflow of the treatment plant than to the outflow. And my question is whether this might indicate the relevance of high flow events bypassing the wastewater treatment plant during, let's say, rainstorms for the ARG patterns in the rivers, that these high flow events are probably more important than the continuous release of treated wastewater. So it is too far interpreted or not?
I'm not sure how these are exactly transmitted. My thought is that the treatment in the treatment plants is not enough, and we observed that previously, and we're not sure if everything is done as it should be in the treatment plants, considering that we have a really low surveillance system. So we intend to find out what's happening.
Okay. Thank you. My name is Anna Baracaracciolo from Italian Water Research Institute. I was very impressed from your presentation and in general from the presentation this morning. And I have just an observation. I understand that this topic is very complicated, but in general, when antibiotic resistance is in a wastewater treatment plant, the general conditions are quite different from the environment in the river, for example. So I wonder if you considered in general the same question to other speakers, the temperature, because I think that once antibiotic-resistant bacteria are in the natural environment, depending on the season, for example, they can find different environmental temperature, very cold, very high, so I think that the temperature can be considered a further stress that can, in such a way, I don't know, because I think that it's a very complex issue to control-select antibiotic-resistant bacteria. What do you think about this point?
As far as I remember from my colleagues that did the experimental work on this, the samples have been isolated twice a year, during the spring and in the winter, I think, okay, or in the summer and in the winter. But I haven't looked on the differences between seasons. We discussed about that, but I didn't have time to look into this particular problem. But I think it's really good to look also, okay, on this aspect.
Thank you.
Thank you.